All right, so good afternoon, everyone. Welcome to module 7.2. We're going to talk about dynamic compartments and Planar polarity. So the idea of this module here is to keep our conversation about cell compartments and how can you use them. But we're going to do something a little bit different now. We're going to have dynamic compartments, and you very soon will see what I mean by that. Okay, so just to recap, in CompuCell3D, and actually in cellular plots model, more general, you can use multiple cell regions to define a cell. So in the first part of the course, every cell was a single blob of collection of pixels, but you can also combine those different pixels to make a more structured cell. And you can have a cluster of cells that makes your real biological cell representation in CompuCell3D, and there are basically two ways to go about it. The first one is using uh, structural compartmentalization, where each uh, cell region corresponds to a specific structure in our cell, like the membrane, the cytoplasm, the nucleus, the mitochondria, some vesicles inside the cell. Or you can also use a structure or functional compartmentalization of a cell, where each cell region or cell domain corresponds to a different region of the cell, which is governed by, it can present a bunch of molecules, proteins, and structures of the cell that has different functionality than the other region of the cell, but that's not a one-to-one correspondence with a specifically organelle or structure in the cell. So we have already seen a few examples of that yesterday morning, and we worked on this model of cell migration where the cell is, you use two compartments to model each cell. One, the yellow one is the nucleus, the purple is the cytoplasm and the membrane, and the green one is the millipodia, where you actually add the forces to make them move. Here on the right is just a three-dimensional version of a functionally compartmentalized cell to model the epithelium. Here I only see a fraction of the epithelium, and in this case, the cells are undergoing now convergent thinning. They're becoming longer in one direction. They're gonna converge and then become thinner and then become longer in the apical basal direction. So one side is the apical side, this the other side is the basal side, and the blue one is, in this particular coloring scheme, is the core center of the cell. And one thing that we did, we only work in 2D, was this kind of, we create this cell epithelium. That was the last exercise that we did on module 6.1, where you create this epithelium made of apical, basal, central cells. And what you're going to do later after my module here is going to add links between the apical side of the cells and induce tissue folding. Okay, so you see that there are many ways you can use compartments, but the way that compartments are usually used in CompuCell or cellular plots models is a little bit, there's some disadvantages on the way that people use it. So one of the disadvantages that actually is the cause all the other ones is that when you do this compartmentalization, you assume there's a fixed position of all the cell compartments relative to one another, right? So usually add the apical side on the top, the basal side on the bottom, right? And the central core in the center, and the cell will tend to stay like that, giving you our choice of addition contact energies. But because they have actually some kind of positive energy interaction between them inside the cell, this means that it's very hard for the cell to rotate or to readapt or change its shape or reposition its compartments if there's any change in the environment. Okay, so that's some kind of disadvantage, because the external environment of the cells may change suddenly, and then if you use compartmentalized cell, the standard technique, the cell cannot quickly adapt. The cell going to be stuck with the apical side at the top and the basal side on the bottom. And if you have a very big cell, it's going to be take even longer for the cell to rotate and readapt or refine its new position. and you are talking about a bunch of cells that have to do it at the time, then you have even then things become even worse because now all the cells have to change, readapt them at the same time, and there's going to be interference between one cell and the others. The other disadvantage in terms of modeling is that you must specify the position of each compartment for every cell that you initialize in your model. And that can be tedious. If you have just one cell to begin with, that's a little bit easier. You just place all of them together. If you have many cells, like the epithelium, becomes a little bit more challenging. In the example that we used last time, we take advantage of the fact that the cells are nicely lined up in the grid. But sometimes you want to have something a little bit more complicated with a different geometry, and then it becomes really a pain to really draw the initial conditions of your model. So that's another practical disadvantage of the current way that we use compartments. The other ones is that Cluster mitosis plugin that we use to divide the cells just copy the compartments relative positions for the two daughter cells, okay? And that might not be ideal in every case. If you have an epithelial cell like this, and you divide the cell along the major axis, now you have a mini epithelium on the top and a mini epithelium on the bottom, with the apical and basal side kind of touching in between. So that's not what it's supposed to be. Then the cell have to kind of find a new position. But because you already start like that, it's gonna take a while and then maybe your cell gonna just kind of flip and become sideways, and then you can get a lot of problems because the relative positions of your compartments are fixed inside the cell. And the other thing is that usually that's not how division happens in epithelial cells. The cell first lose the polarity, divide, and then restart with the polarity and find and those different proteins kind of find their way according to the neighbors or the external environment. So that makes things even worse. Like, yeah, you're not only doing wrong, you're doing wrong, and that in a way that does not help you in the model and also does not affect the biology. Okay, so the division is a problem. Okay, so a way to overcome these challenges is actually using dynamic compartments technique in your CompuCell3D simulations. And the rationale with this technique is very simple. You just have the compartment to the idea is to have the compartments to be dynamic, so they can self-sort out according to current environment. So the current environment is one way, and all the subcompartments, they automatically rearrange to correspond to the environment, and then the environment changes, then the compartments change automatically because of the way you model the cell. So that's why it's called dynamic compartments, okay? So the advantage of this is that if you can have a model like that, you don't need to specify the compartment positions. At the initial, you just start everything fixed, and the cell gonna sort itself out. The cells can quickly adapt to change environment. The cluster mitosis still work, and then you also have the advantage that no matter how you divide, the compartments of the two other cells are gonna self-sort out according to the environment, so gonna very quickly adapt. And you can also use technique to implement the decompartmentalization short before the division, divide, and then recompartmentalize the cell. And that can be a better mimic of the division process. However, this part requires a little bit more coding. You have to convert all your cell compartments to a neutral cell type, divide the cell, and then reconvert the other compartments into the distinct ones, and then you're going to do self-sort out. So that's the rationale and advantage. But then the question, okay, that sounds very good, but how can I make my cell compartments dynamic in CompuCell3D? And the trick is very simple, right? It's actually something that we discussed a little bit yesterday, because some of you asked, okay, what happens if those internal compartment energies become too low, right? And the answer is that if they become too low, then those compartments start to mix one another, right? And that sounds like that's a bad idea because the cell compartments should not mix, especially if you have structural compartmentalization, right? But when you have functional compartmentalization, where each compartment represents a set of proteins, right, that can diffuse inside the cell and self-sort out according to the environment, then this idea of having all them to be otherwise mixed inside the cell is not such a bad idea. Actually, it corresponds more closely to what really happens in the cells, right? In the absence of any external stimuli, many proteins just remains in a diffusive state inside the cell mix. And once there's some kind of external signal or external contact interaction, then these proteins may start to accumulate at the specific side of the cell. Okay, and the way to again induce this mixed internal state of all your compartments is by just assigning negative uh, addition values or contact energies to all your internal compartments. So when they are negative, then it's much better for this compartment to stay mixed with other cells and become clustered together. So you end up from a situation like you have here on the left, where all the compartments are nicely separated, okay, because they have some kind of non-negative addition contact energy between the cells. But as soon as you make them all negative, they want to mix, because that's going to minimize even more the energy. And then you have this situation here where everything's mixed inside. Okay. So that's how you mix the cells inside the cell. And then, okay, but how can this be any good? How can this sort out according to the environment, right? Okay, the next step is you still use positive values for your external contact energies. And it is these positive values of the external contact energies that's going to make these compartments to self-sort out uh, in accordance with whatever is happening outside the cell. So you still have positive values here, right? And it then can be in such a way that all these green compartments like to interact with other green compartments from different cells, so they all sort here. Or maybe you can have some kind of homeotypic positive interaction, where, for example, the red and the green compartments, like this uh, part here is positive but lower than the rest. Therefore, whenever two cells come into contact, you form a green-blue boundary between the cells, and that can be. There are some proteins that actually interact like this. Okay, so so what does these compartments represent? They basically represent any set of proteins that localize a specific cell region as a result from a direct interaction with the external environment. This external environment may be other cells, the ACM, another organ, maybe another agent, maybe a fluid that the cells become in contact with. Can be anything external to the cell that interacts directly with the cell to contact. Okay, and then that can cause its mixing. You can also have these compartments to actually chemotax towards a gradient, so you can have also external gradient. And in the presence of this external gradient, some group of cells kind of move towards that, so they go to one side of the cell or the opposite side of the cell. That's going to be also a way to mix them. So that's going to be another way to polarize the cell of they mix a group of dynamic compartment that will otherwise be mixed inside the cell. So we can also use chemical gradients to do that. But for everything they're going to do today is going to be direct cell contact. Okay, and it can be either between two different cells, or it can be with the medium. If your medium represents the ECM, for example, and your red compartment represents the integrins, then as soon as your cell is surrounded by medium, this which represents the ECM, then this red compartment localized to the membrane of the cell, because integrins is what mediate the interaction of the cell with the ECM. So you can also have that, and you can combine all this together and have multiple compartments inside the cell. There is no limit to how many compartments you can have the cell, as long as your cell is big enough to uh, host all of those compartments. Okay. So when have this ever been used, CompuCell3D? Yes, a few times. I mentioned that before in this model that I did for the Renal-tubule. I'm using compartmentalized cells, so each one of those cells made of apical in green, basal in dark red, cytoplasm in yellow, and red lateral compartment. And you see that if the cell is isolated, these compartments are mixed inside the cell. but as soon as the cell is in a environment where it interacts with the other cells, the lumen and the ACM, then these compartments sort out. And that's much more advanced because I don't have to worry about when I create new cells or growing the cells. I don't have to worry about place the compartments in the correct place. The compartments automatically sort out to the correct positions. So this is a case where I did this for both the renal tube and the cysts that grow out of it. Okay. Uh, Maybe here you can see a movie. So you see a single cell that's growing and forming different cysts, and you can see that all compartments are mixed, okay? And this is the cross section, this is a 3D view. And then you see that as they have more cells and it's forming a lumen, then all the squares start to sort automatically. And you can already appreciate that if I have to do that with static compartments, that would be much harder to do because I have to, after that vision, have to kind of make a loop over what the cell environment is, where I should place the basal side, where I should place the apical side. Well, there's going to be no solution for that. But if I have dynamic compartments, I don't have to worry about it. The cell going to sort itself out. And so the model is more intelligent, it's smarter this way, okay, and makes my life easier. Okay. So, and another one is this somatogenesis project with the cells also. Uh, the things happen too fast here at the beginning of the movie, but I create all my cells with a mixed compartment inside, and they just self-sort out to form these epithelial balls with the basal outside, the apical inside. And in this case, I'm combining that also with epi links to make the cells elongate and also apically constrict uh, in the from the center of the movie. And another example that you're going to be doing today is to model planar cell polarity. And in this case, you're also using an epithelial cell, but you're looking only at the apical surface of the cell on the top here, where you have these proteins, Van Gogh, Brico, Frizzle, Dishevel, Diego, that are otherwise mixed at the cell, but once they see some kind of signal or interact with other cells, they sort out the red to the left, the green to the right, or the proximal and distal, if you use the correct terms. And you see that because of this interaction between them, I'm using the dynamic compartments technique here, so they are otherwise all mixed, but they self-sort out in such a way that they find global orientation uh, after a few Monte Carlo steps of interaction. So they start like this. There's absolutely no external input. The conditions here are periodic. And if I just play this movie again, you see that they all start mixed. Very soon they start sort out, but we have some reds to the top, some reds to the bottom, some reds to the left, some reds to the right. So the polarity of the cell is each each cell is polarized, but they're not aligned with anyone else. And as soon as I let the interaction goes, all the cells start to align with one another and the tissue becomes globally polarized. And in this particular example, it polarized from left to right. Okay, just like in this picture here. Okay, so that's what we're going to be doing today. Before I move on to the first exercise, is there any questions? No? So if there are no questions, then let's move on to the first exercise. And the idea here is to recreate the image that you see on the left panels of the figure. So this on A and C, okay? So let's go work on through that. Okay, so the first thing I want you to do is to go to the cell materials folder and load the simulation template 7.2.1.zip, okay? And then we're gonna have a basic model of a cell with compartments. And then you're gonna just work out how to choose the contact energies to make the compartments do these two outputs, okay? So if you just don't want to code yourself, there's going to be a file 7.2.1a or b, where you already has a solution for this exercise. So if you don't want to code, it's okay. You just follow along with the slides and try to think through the questions that I ask, and you should be able to take to also learn the technique as well. Okay. So if you all have downloaded this file and unzip it and open the folder, the file and read it, you may see that you have, as expected, a contact and contact internal plugins, right? And you may notice that we are using that rule of thumb that we discussed last time, that the contact energies are usually higher than the internal contact energies. Remember, that's necessary to make sure that the cells, your compartmentalized cells have some integrity, right? So if you don't want your cluster of cell compartments to dissolve and mix with the others, it's bad to have the internal energies to be lower than the external contact energies. And you see this happening here because all these energies here are 10 and all these energies here are 2. And I made 0 between likewise compartments because they don't interact. You don't only have one copy of compartment inside each cell. Okay. All right, so that are the energies. If you look at the uniform initializer, you may notice that I start with just a single cell of type C. So in this, in this simulation, we have 3 types of cells: C, B, R, G. So B stands for blue, R stands for red, G stands for green, and the C would stand for cytoplasm. Okay, so that could be the yellow cell that you see in the images. Okay, and you see that you only start with cells of type C. Okay, so all cells are type C. So you can ask, okay, where the other compartments coming from? Okay, the compartmentalization of the cell happens at the Python level in the start function. So if I go now to the steppables file and look at the start, you're gonna see this code here, okay? And what I do here, I have a loop over all cells of type C, okay? And inside, I create all those missing compartments, the blue, the red, and the green, using this command here. I'm not sure you have seen this command to create cells. Did you learn that before or not? Well, I have not seen that before. There is a way for you to draw the cells directly from the Python level. There is a command there, seen before. Okay, great. Where you just can select draw cell, create new cell, and then you have this. You first create the cell. So this is the command that actually creates the cell object, but you have to still place the pixel of the cell to draw the cells in the lattice, right? Before I do that, the first thing I do, I just grab the cluster ID of the cell and reassign to the B, R, and G new cells. Okay, so they are part of the same cluster. And then what I do, I retrieve a list of all the pixels that belong to this cell. Okay, so self get cell pixels list, I got it. And then I go over all the pixels inside it, okay, doing this loop. And then I get the X and Y coordinates of each one of those pixels by use this command here. Pixel tracker data dot pixel dot X, which gives the X position of the pixel, and pixel tracker data dot pixel dot Y, which gives the Y position of the pixel. Remember, these are 2D simulations, so I don't have to worry about the Z. Okay. And then what I do here, I select randomly one of those 6 numbers in this list, okay? And if this number is 1, I assign to that pixel that we are going through to be, okay, that pixel is going to belong to the B cell. If the R equals 2, I say that the pixel is going to belong to the R cell, the red cell. And if this pixel, and if this random number is 3, I assign that pixel to the green cell. So because I'm going over all the pixels and I'm doing this with a random number generator, I have a mixture of blue, red, and green cells. Okay. And what happens if I select a random number that's 4, 5, or 6? Then I do nothing. The cell remains as its original type. It's going to be the type C cytoplasm. Why 6 choices? Okay. It's just because that sets the ratio between cytoplasm and the others. I want to have more cytoplasm than red, green, and blue cells. So that's why I made 50% of my cell to be cytoplasm and that 50% to be these other cell types. So that's a completely arbitrary choice. We may have just make this 1, 2, 3, and 4, and then they're going to be equally divided. Okay. Or I could have increased the one of the populations if I want. So this is completely arbitrary. It's just that's gonna look a little bit better. Okay, thanks for the question. Yes, I'm only changing 50% of the pixels. Exactly. Okay. So after I do that, what I have to remember, right, is that I have to assign a target volume and a lambda volume to all the cells. And this will be whatever it ends up being by giving a random choice. I just set the B target volume to be whatever volume it has after this sort, after this random drawing. And the same thing for the red, same thing for the green, and the same thing for the original cell. Okay, whatever it remains a C becomes the target volume of the cell, and I set the lambda volume of each cell to be 2. Okay, so that's just the code. If you don't have any questions, what I'll ask you to do is to just open CompuCell3D player and play the simulation and see what you get. Or maybe before you run simulation, take a look at the code and say, okay, what I expected to happen? Will this stay like this or not? Okay. Then run simulation, see the output, and tell me if the cells stay pixelized as in the initial condition. So QR code to beach ball, yeah, maybe that. Yeah, it kind of looks like QR code now. And if you scan this QR code, what you get is something like a beach ball, right? So you see that the compartments self-sort out because they have self-energy of 0 by default, but they have a energy of 2 between the compartments, so they gonna sort out to like this and from this beach ball. And Ron actually say that correctly. Cells would sort because all the cell pixels are randomly assigned to RB. And no, someone else said Anastasia. Anastasia said that all because their self energies are 0 and the cross energies are 2. So that's exactly what you get. So the next challenge is, okay, let's go back to the code and fix this to mix the compartments. Okay. So let's change the contact energy to mix this. Okay, so I realized that my slide gave up the solution. Okay, so one thing that people can do when they start to try to make the cells remain mixed is they may try to assign 0 to the cells, and some will try to assign negative values. However, if you assign negative, just 0, you do not get mixing. Okay, you still get some kind of clusterization of the compartments. The compartments look much fuzzier than before, but not as mixed as they should be. You have to make your energy to be negative to actually get the cells mixed. Okay, so if you tried 0, you get something like this. But if you try a negative value like -5 or any negative value, then you start to really get mixing. So why does 0 does not do the trick? Does anyone know the answer? Yeah, so yeah, it's not repulsion, it's more like a lack of force, yes, because they're as likely to be the next to themselves than next to another color cell. Yeah, that's true, but why they're more likely to be, say, next to themselves? What causes that? The answer to that, you have to go back to out at the beginning. No, it does not depend on initial arrangement because the initial arrangement is completely random. The reason you have to go back to the intro to pots course that you have seen from Pedro the first week is that the way that you grow pixels is by copying a pixel to another one that's next to it. So because the pixels evolve by a copied attempt of lattice copy attempt that's right next to the original pixel, this makes the pixels position to be always a little bit correlated. Okay, so that's why they tend to be together, even though they in principle have no preference to be with the blue, the red, or the yellow. Okay, it's just that the way that the pixels move inside your compartment is by copying a pixel to a neighboring one. So that's why they becomes correlated. So to overcome that, you actually have to make our contact energy to be negative, and then they will actively mix with the other compartments. Does that make sense? Okay, if it doesn't, or you have a question, let me know. Now let's move to the next exercise. Now I want you to change your values to get all the cytoplasm to be inside. Okay, and I think you have already done something similar on the module 6.1. So so now that there's a challenge, what you have to do for this to happen? Try on your code and let me know if you get it and what you did to get it to work. Click on Anastasia for sharing your output. So at least one of you got it to self-sort inside. So of course, when you do this, you're saying that your yellow compartment corresponds to cytoplasm, and your green, blue, and red compartments are some kind of membrane molecule or some molecule that's say close to the membrane or is directly interacting to the membrane, right? So when you do this, you're already giving some kind of information what type of molecules this red, green, and blue are. Okay. So Anastasia got it sorted to inside. So actually, to get this to work, you have to do two things to get to sort inside. Okay, someone almost got it, but there is some kind of sorting of the blue to one side. So you probably make positive the blue with everything else. Okay. Luis Iago. Okay, Osman got it. So Osman and Anastasia, what do you have to change to get this to work? So let's see what Taylor did. Internal, she made, okay. So look at what Taylor did. So one thing that Taylor did, she made the C and the B, the C and the red and the C and the green to be actually positive, right? So it has a positive energy with the auto compartment. So we want to sort from the auto compartments, okay? So so that's the answer from Taylor that I can see from her screen. Okay, Taylor told me that she also did something else, right? Yeah, so she also changed the energy of the yellow compartment and the medium to be high. Okay. So this is the solution, right? You actually need two steps. Okay. The first thing that you may think, okay, if I want to medium to avoid outside, I just increase the external contact energy of the C when the medium to a very high value, 40 or 50. But that's not enough. That you're just going to prevent the yellow from seeing the membrane, moving away from the outlier of the cell, but you not necessarily make the it to sort from the other compartments, right? So if you just do this, you got this output where all the yellow compartments go inside, they never touch the membrane, right? But you still have mixture of your alter proteins inside the cytoplasm. And that may be a desirable output. Maybe your other proteins, they can be on the membrane and also bulk concentration. So that's going to be a good way to do it. But if you want to completely sort the other compartments to the membrane, you also have to increase you have to make the energy of the cytoplasm and all the other compartments slightly positive so they can also sort out. Okay, so just doing this is not enough. You have to do both to get it sorted out. The other thing that you have to know is that if you just do this part here, there's also not enough for the cytoplasm to go to the center, because the cytoplasm can still segregate from the other compartments, but go to the side of the cell, right? And not necessarily to the center. If you want it to be to the center, you must have a very high contact energy with the external environment, or the medium in this case. So you have to change things both in the external contact energy to add this part here, and internal contact energy to add this self-repulsion between the compartments. And here I want to already introduce you to some notation that I like to use, because now we have a set of contact energies that are external and a set of contact energies that are internal. I like to use capital J to refer to all the external contact energies, and a lowercase j to refer to all the internal contact energies. So that's going to be the naming scheme that I use when I deal with both external and internal contact energies. Okay, that's another rule. That's something that I use as a guide. But I think it's important to use different ways to refer to the contact energy, so it's become very clear when is external and when is internal. So, question from Ava: is sufficient to keep CC at 0 because we increase CCRG and CCRB, or you want CC to be negative? Very good question, because that's something that's very important. The self-interaction of the compartment with itself is always 0, okay? Or any cell with itself is always 0, okay? So, and that's independent of this code that you see here. So this code here is not setting the self energy between C and C to be 0. It's just telling that if you have two cytoplasm cells inside your cluster, they would interact with that energy, okay? But in this case, you only have one copy of each compartment inside the cell. That's why you can pretty much ignore all these energies here. So in other words, if you change this energy to be 10 or 100, there's going to be no effect on the simulation, because you only have one copy of the cytoplasm in your cell, and you only have one copy of the blue compartment, only one copy of the red compartment, and only one copy of the green compartment. Almost like that. Those values only matter if you have two copies of them inside the cell. So if you go back to your example here at the beginning of cell crawling, or maybe if you just go back to this example here, see that you have one nucleus, right? But many cells in biology, they actually have multiple nuclei inside it. So if you have a situation where you have a cell that must have two nucleus, you're going to use two cells, blue cells inside your cluster cell. And in this case, you have to specify what's going to be the interaction between the blue and the blue cell, or the blue and the blue compartment. Okay, that makes sense. So we only have multiple copies, then that becomes so that those diagonal terms in the internal contact energy become relevant. Okay, great. Glad it makes sense. Okay, so let's go back here where you were. Yes, we're here, and then, okay, let's now move to the next task, okay? Just keep the mod that you have, and now make all the red compartments to sort towards the outside. So what you have to do? So the first question I have to you, in order to get this, what do you have to change? Do you have to change the external contact energy or the internal contact energy? Okay, Troy already gave an answer. You have to change the hierarchy where the red cells like the medium more. Okay, you do that in which energy, the internal or the external? Anastasia already gave the answer. Lower external J between medium and R. Yes. So alright, C. Okay, you're doing very well. Okay. That's exactly what you do. So of course the values that you choose are going to be your choice, but if I just choose my external, see the big J here, contact energy between red and medium to be 2, which is going to be lower than the 10 that we use for everything else, then the red goes to the external one. You want to have the low, but you don't want to have it negative, okay? Because if you make this negative, especially if you make lower than -5, the red cell will go to the medium, but the red cell actually gonna diffuse towards the medium. You actually break the red compartment. If you want to try that, make this energy to be -10, and you're gonna see the effect of the red exploding towards the medium. If you want to do that and share a snapshot. So let's move on. So we read this exercise, maybe you already got the gist of how this works, right? But you're only seeing the sorting out of 2 compartments that goes inside. So that's more like making the initial condition of the cell and 1 compartment going outside of the medium. Things becomes more nice when you actually have 2 cells, right? Let's move on to the next exercise where let's try to produce the panels B and D here. Kaya did a very negative R medium contact energy. And then you can see, so if it comes too negative, then the red compartment prefers to be with the medium than with its own compartments. So it just explodes. Okay, so the cluster, when we use cluster, we refer to all the compartments that belong to the same cell, or all the compartments that have the same cluster ID. Okay, the compartments are these cell objects of different colors that you have inside the cluster. Okay, so this will be one cluster of one cell cluster we made of different compartments: red, green, blue, and yellow, and outside is the medium. Here on the right, you have 2 cell clusters, one on the right. Okay, all these compartments here have the same cluster ID, so they belong to the cell on the right. And here you have another cell cluster, and all these compartments inside, they have the same cluster ID, so they belong to the same biological cell representation. I know it's a little bit confusing because initially CompuCell3D was developed for cells without this concept of cell compartments and cluster. So we are still kind of in the code treating this colonies compartment cells, right? But they're not really cells. Okay. Maybe a bad way to define is that you have those domains. You have a red domain, a blue domain, a green domain, and yellow domain, right? And sometimes a single domain corresponds to a cell, and sometimes multiple domains should correspond to a cell. Okay, so that can be the more generic way to refer to this. So when I publish papers, I try to use this nomenclature of using domains, and sometimes a cell is a single domain, and sometimes a cell is a collection of multiple domains. Anastasia, I already want to go to do this exercise. Yes, you have to initialize 2 cells, okay? It's better this way. And to do this is very simple. You just go to the uniform initializer, and instead of having 20 to 40 on the x coordinate, you make just 20 to 68, and now you should have 2 cells side by side. So if you just do this, save and hit enter, and play the simulation, you start with this initial condition, 2 pixelized cells, okay, side by side. That looks very nice. But when you run the simulation, you may get something that looks like this. Did everybody got this kind of output? Yes? Okay. Okay, so now the next task is, okay, let's fix the output. We don't want this to happen. We want this to happen. So now let's change your code to get this output instead of this. What do you have to do? So now is your task. So a tip here, you have to change multiple variables, okay? Multiple values in the external contact plugin. Ah, excellent. Very good, Martin, you got it. Osman actually got it, but I think, Osman, you did something else as well, okay? You ready to move on to the next exercise? Okay, so for this one, what you have to do, you have to make the cytoplasm to not only avoid the medium, but you also have to avoid anything else that does not belong to the cell. So everything that's external to the cell, the cytoplasm should retract. So you have to also change the external energy of the cell, of the cytoplasm with other cytoplasm, okay, so they don't touch, but also do that for the other blue compartments, the other red compartment, the other green compartments that are not themselves. So you have to make all these equal to 40 or 50. And when you do that, then it really separates and you get this output. So did you all got this? And does the solution make sense to you? I got something similar, but I decreased the external energy between green and blue. Okay, in this case, the green and blue like to colocalize with each other, right? But that's not necessarily forcing the cytoplasm to move away. Actually, when you do this, you are getting this output here, right? Something like this. Okay. The more so you write gives some kind of information what the green and the blue means. Yes. Uh, so if before you add this information what the green and the blue proteins are, or the green proteins are, the most general way is just to make it avoid equally all the external regions. So this is going to be the more neutral or null hypothesis solution to the code. I see the reply also got it. Excellent and Honak. Yeah, because you make the red and the green to like each other, you see along their. I look at your image here. First, you maximize the contact between them, and you always have a green and blue pair interaction. Okay, but this is going to be the most general answer because you're not assigning any meaning to the red and the green compartments. Does it make sense? Okay, so once now we have this, now let's try to reproduce those two images here. Okay, one where the green colocalizes the surface, but the red does not go to the outside. Another where you have the red outside and the green blue on the membrane. Well, we already know the result for this second one because both Osman and Anastasia got it. Okay, but probably already know more or less how it works, right? I'm gonna just kind of leave the answers here, and I'm gonna give you a little bit of time for you to complete those two exercises. Maybe you can try your own or just look at here what I did on each one of them and try to reproduce yourself. So I'm gonna give you a 1 minute before we move on to the next exercise, which is going to be modeling planar cell polarity. Yes, you want them to colocalize with one another. So that's going to be a simulation of Fs and F rings. Okay, so these are two transmembrane proteins that do not interact with other of its kind, but only interact with the other kind. So a heterotypic interaction. So F rings on a cell membrane likes to bind to the Fs on the other cell membrane. Okay, so that's what they do. So usually cadering, so and the one on the left is more like cadering. So you have E cadering, it likes to bind to another E cadering on the other side. So if your green compartment represents E caderings, So when you bring two cells together, they kind of localize and bind there and makes the connection. But if your red and blue compartments represent Fs and F rings, then they have this heterotypic interaction, right? Is F with F rings and vice versa. So that's why you get this kind of blue and red region, and another which is red and blue. So this is going to be the cell boundary right in between here. So to do that, you lower the energy between blue and green, or make it 0, so they're going to colocalize here. On the other one, you only lower the energy between green and green, and then they colocalize in the membrane. So I'm gonna move on. Oh, very good, Anastasia. You got all a single red and blue output. So the next exercise, you actually want to try to do the planar cell polarity exercise. So in this case, we're going to start with a bunch of cells in the simulation. And then they're all going to be pixelized with internal cell compartments. All their energy is going to be 0, but they're going to have positive external energies in such a way that the red and green compartments sort out to different sides of the cell. Okay, that's the idea. But I'm not going to give you this code. I'm going to give a code that's only set the initial conditions. Okay, and then it's going to be our task to figure out what kind of energy you have to choose to make this work. Okay, so it's a model design exercise and not really a model building exercise. So to do this, please go to the cell material properties and download the base file 7.2.3.zip, open and see what's inside. Okay, basically should be a template simulation that's similar to what we had before. We have 4 cell types: cytoplasm, PD, which stands for proximal and distal. And proximal is going to be the red, distal is going to be the green. Okay. All the cells start as a type C, and then there's a routine inside the start function that pixelates all the cell or add the compartments just like what we have done before. Okay. And then all the external energy starts at 10, and all the internal energy start at 0. Okay. So, all right, telling more or less what's inside the code. Please download it, unzip, open tweet it, and play, and tell me what you get with this file. Okay, Nastasia got a mess, right? Mika also got a mess. So, Mika, I have a suggestion for you. To better visualize what belongs to the same cell, what belongs to different cell, or what belongs to the same cluster, what belongs to different cluster, you can go to the visualization in the player, and under visualization you have an option of cell borders and cell cluster borders. Select only the cell cluster borders, so unselect the cell borders, and when you do this you get an output that looks a little bit more like what Anastasia has, where you don't draw the boundaries between the compartments. Okay, so that's gonna aid your visualization. And the same suggestion I also give to Karini. You can go to the visualization and have only the cell cluster boundaries selected and unselect the cell border. Some said these are Pollock paintings, but usually he does more like lines, right? Yeah, it's a mixture of Pollock and Mondrian. Okay, so if you all don't know the code, right, you should get something that looks like this. Each star out cells starts square root, right? Then I pixelate the cells, and the very first Monte Carlo step, you may see something that looks like this, and eventually they sort out, but there's absolutely no pattern emerging. Everything is still a mess, even though the two compartments sort out. And that happens because all our external energies are 10 and all our internal energies are 0. But that's not the output that you want to have. The output that you want is something that looks like this, okay? So take a look at this output. What you want to have, right? This is what you want to have, but this is what we have because we have no information about the contact internal and external. So our first task is let's change these values to make a planar polarity type of simulation, okay? And again, we have to first start with a qualitative choice for the different energies, okay? So let's do this exercise again, okay? It's a model design exercise. I don't have read done this before, but let's practice once again, and let's see if you can get this to work. So let's start with the contact internal, okay? What we want for the contact internal energies to be like, okay? So right now they're all 0, okay? And I already told you that we don't have one copy of each compartment inside the cell, so we can ignore these diagonal terms. But what about these off-diagonal terms here? We have proximal, distal, and lateral, and the cytoplasm as well. What they should be? Right now they're all 0. Should you modify any one of them or not? Let me know in the chat. Okay, I got 2 answers so far. We want red and green to be larger than each with lateral. Maybe. Why, Clara, you want that? Anastasia suggests cyto should be high with everyone else. You can do that, but why? And then she suggested P and D, or the red and green, to also be high with each other. So Anastasia and Clara, why do you want that? And Anastasia, why cyto should be high with everyone else? So cyto is on the inside by itself. Okay. And the red and green on the proximal distal, PD, why they should be a little bit higher? Don't want not want them to like each other, so they're gonna physically separate. Yes. Oh, and then Clara already went further. She said that in the external one, they should like each other. Okay, yes, that's true. Okay, so very good suggestions. When it comes to planar polarity, right, there's a few different choices you can make. You can certainly make the cytoplasm to be high, so they're gonna go outside. So that's a good suggestion, right? So that's completely valid. And you may want to make the proximal distal to actually dislike each other, so gonna internally segregate to different sizes of tissue. That's also a good choice. But none of them are really necessary, okay? So here I just make this choice that the proximal distal, they do segregate internally. And in real biological systems, sometimes this happens, sometimes it does not happen. When you talk about the Drosophila cells in the wing, the Drosophila, okay, now I don't remember details, but in mammalian systems and in non-mammalian systems like the Drosophila, the fruit fly, sometimes the cell has a capacity to self-segregate the proximal distal by itself, and sometimes the cell does not have this property of self-segregate. So it's different between mice and fruit flies. So in one of them, you want to model with a internal repulsion between proximal distal, because that's what you see in experiments. When you isolate the cells, they do segregate. And sometimes, and in other systems, when you do isolate the cells, you don't see the segregation. So depending on whether you talk with modeling planar polarity in mice or modeling planar polarity in fruit flies, you may want this have this energy to be positive, or maybe you want leave this energy to be 0 or even negative. Okay, so it really depends on biological system, but both answers are correct. Okay, now which one is which, I don't remember by now, but there is a difference between mammalian and fruit flies in that respect. Okay, and then you can make the cytoplasm to go to the center and now then to be outside. These ones are a little bit more unsure about that because usually all those proteins do stay on the apical surface of the cell. So we're talking about the apical surface. So they are already in the membrane somehow. So you may do that, you may not do that. It really depends. Maybe your proteins are more subapical than really apical. And then again, there's going to be a choice. You can make that to be a little bit positive or not. But all the answers that you gave are correct in some sense. They are going to reflect different biologies, okay? Different systems. But this is going to be the choice that I made. I have alpha in zero. Maybe I should just make them negative to win this mixing. That may give better results, actually. But there's going to be one hierarchy that we want to do, okay? Let's go to the other side. Okay, so now let's go to the external contact energies. So what's the very first thing that you want to do in the external contact energies that you need to, it must be true in order for this tissue level organization now to happen? Okay, Replus suggests that maybe the cytoplasm should stay away from each other from the medium. So, okay, in this model, there is no medium, so don't have to worry about it because it's periodic boundary conditions. So already simplified the things for you. No medium in this model. But Replus suggests maybe CC should be high. Yes, that is a solution. Actually, I kind of add us. I didn't define the CC in this set of slides. I should fix that. Yeah, but you can have CC high so they stay inside. What else do you want? Okay, Clara suggests 0 P and D. Okay, proximal distal should be 0. Should it be 0 or should it be just low? No. Okay, Replus said PP, TD, and LL should be low. Okay. So yes, you want the P and D to be low because you want them to colocalize externally between two cells, right? So you certainly want to do this. So let's just go ahead and write low here. And then I think Replus said that the cytoplasm should stay away from each other for the other cell compartments. And so maybe it should be high. So that's actually my next slide. Let's make the cytoplasm high with everything else. Okay. So I don't have the coupling of the external proximal distal compartments or the lateral compartments to the outside. By the way, when I say lateral compartments, I mean the compartment they're going to kind of localize across the cell on these boundaries here, okay? Just to be clear what I mean by lateral. So what about proximal, and distal? So one of you said they should be low. Do you all agree that those areas should also be low or not? Some said that should be high. Why high? So some of you said that PP and DD should be high, but PD should be low. Okay, why do you want this hierarchy? Clara said fixed orientation, but how can this fix the orientation? Okay. We don't want PP to colocalize. They need to be on the opposite sides of the cells. Do you agree with that? Yeah, that's exactly true. And the same thing goes for the DD. You want them to be high because you don't want to have red and red colocalization and green colocalization. So you want those two terms here to be high. Maybe not as high as the cytoplasm and the other ones, but it should be relatively high. Do you agree? Does this make sense? Okay, so what about the latter one? So what about the lateral? What value should you use here? Low, high, very high, or medium? Yeah, okay. So why should it be low? So they can be adjacent. Yes, good answer. But should they be as low as P and D? Or they should be a little bit higher, or should be even lower? So I'll give you my answer. I set this to be all medium, both LL and also LD and LP. So do you know why I did this? I suggest doing this. It's basically because this lateral compartment is just the absence of the proximal and distal proteins that drive the process. So this is a more neutral cell region that's not driving the compartmentalization and or the polarity of the cell. So I don't want the lateral to active colocalize for another atom because then again saying that there is active mechanism for that. Okay. So I want it to be neutral between themselves and also be neutral with the other ones. So this way I assign no active property of this whatever proteins are in the lateral one. It's more the absence. The action happens with the proximal distal proteins, not on the lateral ones that have nothing. So maybe you can see maybe there have cadmiums there, and then maybe lateral should actually be a little bit lower, but I suggest doing them all medium because then it's a completely neutral boundary with respect to everything else. Okay. Okay. So there was a question. If CC is high, do you need to set CP, CD, and CL as high? If you want the cytoplasm to be inside, then you also need that. Maybe CD and CL does need to be, and CP does need to be as high as CC, but you probably want to be higher so they couldn't be inside each cell and never touch each other. Okay. So the other compartments are really the membrane compartments. Okay. But anyway, I chose this set of values for my lateral compartments. Again, you may change this depending on the biology. So many of those things you can actually change based on the biology. So some things have to be, this has to be low, this has to be high. That's all set, okay? These may be medium, high, or maybe negative, okay? So some of these values are, you need a hierarchy for it to work. Some of the other values may be changed slightly depending on the biology. But the idea is you do this qualitative design of the model, and once you do this, then you turn into numerical values, okay? And I'm already finishing now. So this is just one example of how I choose my numbers. Okay, you can choose another set of numbers. Maybe you probably can do better than this. My student that works on a model of Proximal polarity, she ran a lot of simulations to a parameter scan of all simulations to maximize the polarization of the cells, and she came up with different values that are more optimal for this to happen. But if you use the ones that I show here, you end up getting something that looks like this, as in the where you see all the reds go to the down to the left, and all the greens more go to the up and to the right. So you have some kind of polarity happening, but you can make your own choice of the adhesion values. Okay. Well, the only thing that I have left is actually to visualize this using the vector field. So right now, what you can see, okay, if you look at each one, say, okay, the red is here, the green is here, so there's a polarity in this direction. If you look at the other cell, there's a red on the top and the green on the bottom, so there's a polarity in this direction. But you see that just looking at the cells, all the information is there, but it's kind of hard to read by the eyes, right? You have to stay a very long time looking at all those cells and figure out, okay, where is the direction, okay? But it would be much easier if you actually could convert all these images into a little arrow that points from the red and the green. Okay, do you agree with that? Okay, I hope you do. Okay, because that's the what I want to do next. Okay, so this is something that we can go together, or you can just download the final file and have the vector field already implemented in the code. So I'm gonna go slightly quick here, just to show how this is done. So basically what you want to do, you want to have the cell field visualization, you want to have another window of visualization in your CompuCell3D player, where instead of displaying the cells, you replace each cell by a vector, by an arrow, that's gonna show the direction from red to green for each one of those cells. Okay, so to do this, we can go to the code, and you write again, you're gonna add a steppable. So you go to the steppable files, right click and select the add steppable file. We did that yesterday. And here I want to do two changes. First, give a steppable name, let's call cell orientation, because it's gonna be the orientation of each cell. Leave it generic. Select a frequency of 10, so your code does not becomes too your code runs fast, okay? So you're going to do this every 10 Monte Carlo steps. So that's the period at which you do this calculation or the frequency. And here on the bottom, you have those options that we never used before. And we're going to want to use the option vector at the cell level, which means that every cell is going to get a vector, okay? And that's exactly what you want. So you're going to select vector cell level. You hit OK, okay? And then CompuCell is gonna, the tweet is gonna place a code for you, okay, with the vector field, and this code is very generic, okay. And what you want to do, you want to modify the code to do what you want to do. So let me go to the first part here. First, you want to import all the math library, okay. This is always a good idea. I think that should be the default on CompuCell Python code. So I will always import math and NumPy. Okay, and then here on this first line here, where you create the vector field cell level Python, you have to give a name for the vector field. So let's call it cell orientation as well, okay? Or PCP orientation, that's going to be also a good name. And here, what I want to do, I want to add this line here, self.field equals, and then this comment here, because then you store the field as a variable that I can access in the step function. So that's what I want to do here. Again, don't worry about coding this. I'm just showing this. And I have the file with all this already coded for you at the end. I want you to follow along. So here you see that the frequency is 10. There's the name of the steppable. I give a name to the field, and I store the field as a variable with the self dot in front so that I can access it within my step function. Then on the step function, what I want to do, I want to loop over all the cells and calculate the vector that connects the proximal compartment to the distal compartment, or the red to the green compartments. So I do a list over all the cytoplasm cells, okay? And then I get the lists of all the compartments, okay? So I give the cell cluster ID and I get a list of the compartments that belong to that cluster. And then I set P variable and D variable to be 0, and I go over all those compartments. And once I get the compartment that's of type proximal, I store that in a variable P. And once I get the cell that is of type D, or the distal compartment, I store that in the variable D. So this is storing the components and variables. Okay, so now I'm looping over all the cytoplasm cells, and then from that I'm getting the proximal and distal compartment and storing them. And now once I do that, I can now go ahead and use that common self-invariant distance vector between cells, and then I give P and D as the two cells that I'm calculating the distance, and I get the vector components of the distance between them. And with these vector components, what I can do, I can, again, calculate the angle with respect to the x-axis using the A-tangent function and given y and x, as we did before for the cell crawling example. And then I reconvert that into another vector again. And it seems that I kind of wasting a little bit of time. Why am I doing this? I'm doing this just that my vector will all have be unit vectors. Okay, so that's a little bit of math here. I want all the vectors that I display to be the same size. So I get the angle and then use cosine and sine to get back then as unit vector on x, y, and z. And I store that in the list where the first enter the list is going to be the x length vector, the second position, the y length vector, and the last position is going to be the z length vector, in this case is 0 because it's a two-dimensional simulations. So now that I get this vector, then I populate my vector field with this vector. So I have this vector field, cell field that I store right at the beginning here. Remember, this is going to be the field that we created, and I store that as a variable. And then I populate this variable, so I give as an entry the cell which I'm referring to, and then the vector that belongs to that cell. Okay, so that's gonna, for each cluster, I'm gonna draw a vector that connects the proximal compartment and this compartment. So I'm gonna give the unit vector there. Okay, so after I do this, so this is the whole code for you to see, right? Create the field, give a name, go over all the cells, get the compartment set cells, grab the proximal of this compartment, and then if I have a proximal of this compartment, this is just a check. I get a vector, distance between them, I normalize it, and I use that to populate the vector field. Okay, so once you have that and run, and you can just download this coded from file 7.2.3b, right? And you run simulation, then this is what you're gonna get. You're gonna get, in addition to your cell field, you also have a sector vector field where you can see the orientation of its cells, and you can appreciate that this is much easier to see or to interpret by your eyes than this one, okay? So that's a different way to visualize. So when you download this, unzip and run simulation, okay, you automatically get only this field, and we ask you, how can I get this other field, right? There are two ways to do this. You can click on these arrows here and you can select these other fields. So remember that you have a vector field, a scalar field, a vector-based field, and a scalar cell-based field. You can create as many fields as you want, and you can select them here. But then you're gonna lose the cell field. If you want to see both of them at the same time, what you can do, you can click on this button here in the player. That's gonna create a new visualization window, okay? So that creates a new window, and now when you click this, you're gonna have two cell windows side by side, and then the one on the right, you can click here and select the field that you want to see. And then in this case, you select the cell orientation field, with the name that you gave to it, and then you're gonna see all those arrows displayed for you. So I know that I went a little bit quick on this, how do you create a vector visual field, but you're gonna have a dedicated class to explain all of them tomorrow. But here's the code, 7.2.3.zip. You can open and see in at your own pace exactly how this was coded. Okay, but that is the end for this module for now. I again thank you for your attention, and please give the feedback with the questionnaire.